So the last subtopic of the biochemistry option is biochemistry in the environment. It's a little bit different than the other subtopics that really focused on different categories of biological molecules and mainly considering how they behave within animals and humans in particular. This one is going to look at some of the effects on other species in the environment of pollutants, and also some of the sort of approaches that people have taken to clean up pollutants and to remediate some of the problems of pollution and how those involve biochemical molecules. It's also going to look a little bit at the philosophy of green chemistry and some examples of that. So to start off, we're going to talk about xenobiotics. So like xenophobia. Xenophobia means you're afraid of foreigners. Well, xenobiotics are sort of foreign chemicals to an organism. So usually they are human-made synthetic chemicals, or they could also be natural chemicals that are occurring at abnormally high concentrations, and we'll see an example of that later on. But these things could be like antibiotic molecules, hormone molecules from pharmaceuticals that people have taken, and that don't get fully processed. So either they get thrown out, they're waste products from the drug companies, or even sometimes some of them don't get metabolized in your body and you end up urinating them out. And so they can end up in the environment that way. Insecticides, so chemical, pesticides that have been sprayed like DDT, pollutants such as PCBs, dioxins. These are, lots of different examples of chemicals that make their way into the environment and then they can have very negative consequences. So metabolized or not is a big issue. Most organisms have enzymes that are designed to help break down foreign invaders, foreign molecules. But xenobiotic, if that's the case, then that xenobiotic is metabolized. But a lot of the times, because these are human-made molecules, the animal may not have an enzyme that's evolved to fit with that xenobiotic molecule. And so it can't break it down and it can't get metabolized. That's often because of carbon to halogen bonds or aromatic structures, meaning containing phenyl groups, benzene rings. Those are structures that don't occur very often in biological molecules, and often they're pretty strongly bonded together. And so there's not many enzymes that can easily break those down. Two terms you need to know and be able to distinguish between is bioaccumulation and biomagnification. If a xenobiotic makes it into the environment and it doesn't get metabolized because it's relatively chemically stable and unreactive, it will accumulate in that organism over time. A lot of these xenobiotics end up being fairly nonpolar in their structure, and so they can get through cell membranes, which are lipid-based, and they're also fat-soluble, so they end up getting stored into fatty tissues. So this happens with things like methylmercury and tetraethyl lead, which comes from leaded gasoline. So over time, because they're not really being broken down, more and more of this xenobiotic, this pollutant, will store up in the fatty tissues of that animal. Now, biomagnification is a little bit different. This refers to when bioaccumulation is happening over an entire food web. So the idea that you've got these different trophic levels of a food web, starting with little producers of energy like the little tiny phytoplankton and algae that produce energy using photosynthesis. And then you've got the tiny creatures that eat them. You've got the small fish that eat them, or whatever the animal is. I'm using fish as the example. Then you've got the bigger fish, and then you've got the top predators at the top of the food chain. With each one of those steps, as you go higher up the food chain, the concentration of that pollutant, of that xenobiotic, is going to get higher and higher and higher, because every level up, that creature is eating a lot of the smaller creatures in order to survive. And so, creatures at the top of the food chain, like an eagle, or in our case, humans, we are pretty much the top of the food chain. And so when there are pollutants in the environment that get into creatures, we end up with fairly high levels of these pollutants in us. And so that concept that the higher you are in the food chain, the more concentrated that pollutant is going to show up, that is called biomagnification. So bioaccumulation is one organism and biomagnification is an entire food web. A couple of specific xenobiotics to know. We've got dichloro-diphenyl trichloroethane. That's a IUPAC name. It's quite a mouthful, but you can sort of see that you've got dichloro, so that refers to these two chloros. Then you've got diphenyl, which refers to these two benzene rings. And then you've got trichloroethane, which is this part of the molecule here. You've got the two carbons, one there and one there, and then the trichloro. So that's where the name comes from. DDT is rather famous. It was used in World War II to help kill mosquitoes, prevent malaria in the troops, and it saved millions of lives at the time. But as it got more and more concentrated in the food webs, it ended up really affecting birds, especially big predator birds at the top of the food chain, because it was stored up at high levels through biomagnification, and it ended up making their eggs very thin. And then when the parent birds would sit on the eggs, they would crush the eggs, and so obviously they didn't have any babies. And so for a while, the numbers for these birds was drastically dropping because the numbers of offspring was really going down because of this pollutant. There's a lot of other xenobiotics. The examples, I already mentioned this before, that things from the pharmaceutical industry like antibiotics, this is a structure of penicillin, or hormones like female sex hormones that would be in the birth control pill. They often make their way, through the human body, not completely metabolized, not completely broken down by bacteria, and they end up in the environment. Other drugs like painkillers and chemotherapy drugs can also do this. The female sex hormone ones are interesting because they can have effects on male creatures and make them actually stop acting entirely like males. This is especially true for fish, I'm sure other creatures, but the example I can think of is fish. And the males start to have more female characteristics or hermaphroditic characteristics as a result of high levels of female sex hormones in the water. There's actually another category of pollutants that are called xenoestrogens. So these are chemicals that, although they're not actually female sex hormones, they imitate the effects of them. So they imitate the effects of what we call the estrogen group of compounds. So these include the PCBs, which are substances called polychlorinated biphenyls. You can probably guess why that's the name. Many chlorine atoms off of two phenyl groups. They're a pollutant released in the electronics industry. And bisphenol A, more famously known as BPA, which is a plastic additive, again, also has a somewhat similar structure to estradiol, and it can have these estrogen-like effects on creatures. Okay, so then there's lots of parts of this biochemistry topic that have to do with amelioration or improving these problems and outlooks from the xenobiotics. One category is the host-guest chemistry. This is the idea of trying to mimic enzyme-substrate interactions in order to remove a pollutant. The idea is you have a molecule that you've designed, a synthetic molecule that we call the host, and it's designed to sort of fit with and bind with some sort of xenobiotic molecule or ion, which is called the guest. And the key is that these attractions are going to be non-covalent attractions, so that these things can be temporary. So the attractions that could exist between the host and the guest are London forces, hydrogen bonds, dipole-dipole forces, and ionic bonds, like we've talked about in the protein topic. And then as they are bound together, it's referred to as the host-guest complex, and it can be called a supramolecule. It's not really an actual molecule, right? It's these two things sort of temporarily bound together, but think of it almost as like a supermolecule. So it's called a supramolecule. And it's similar to how the enzyme and substrate bind together temporarily in the enzyme-substrate complex. The host and the guest are, the host is often considered to be like a cage or like a tube, and the xenobiotic is designed to fit inside. This is used effectively against collecting cesium ions that are radioactive and also carcinogenic amines in the cosmetic industry. Another way to try to improve the outlook for xenobiotics is to make substances more biodegradable. So this focuses on plastics. And so there are two forms of biodegradable plastics that have been invented, and they're generally more able to be broken down because plastics tend to, at least made through addition polymerization, tend to have a lot of alkene bonds, so just carbon to carbon bonds, which are pretty strong. And so these other kinds have a lot of groups like carboxylic acid groups or hydroxyl groups or ester groups, which are a little bit easier for a lot of enzymes to break down. So one form of biodegradable plastic is plant-based hydro-biodegradable plastics. It has a high starch content. It's usually made from corn, but they're developing ways to make it from grass as well. Part of how it works is it contains a lot of small starch pieces that when they get wet, they actually absorb the water and expand, which helps to break the plastic down into very small pieces. And then further hydrolysis reactions take place naturally to break down those molecules, and they produce carbon dioxide and water. And these reactions are aided by high temperatures. If you bury these plastics in a landfill, however, sometimes they are still very slow to break down, and they can lead to methane production since there's not enough oxygen buried deep underground. And methane has its own negative effects because it's a very potent greenhouse gas. The other kind of biodegradable plastic is actually made from a byproduct of the oil industry. So it's using something that would otherwise go to waste. It actually contains cobalt as part of its structure, and the cobalt acts as like this little catalyst site for the breaking apart of the plastic into smaller pieces. And then bacteria contain enzymes that can break down these pieces once they are small enough. Another example of trying to use enzymes to bioremediate a situation, cleaning up industrial waste with enzymes. It's used in different industries, but a specific one that's talked about in the textbook is cleaning up oil spills. So crude oil is a natural substance, but it can be considered a xenobiotic when its concentration is abnormally high, like with an oil spill. And so using a mixture of different bacteria and fungi, there's usually the right cocktail of enzymes that can actually break down all or most of the components of crude oil. And so while it's really dependent on how warm the bacteria and fungi are, if they have enough nutrients, if they have enough oxygen, they usually can clean up an oil spill given enough time. It's usually not fast enough to prevent ecological damage from an oil spill. However, it is still a way to eventually clean up a lot of the oil that's in the water after an oil spill. So using enzymes from bacteria and fungi to clean up something like an oil spill. And the last topics are these fundamentals of these principles of green energy. There's lots of different ones, but pretty much it has to do with being more environmentally friendly. So trying to produce less waste, using less harmful toxic solvents and chemicals, using more energy efficient processes, using reactions that have greater atom economy. The equation for atom economy is in the data booklet. It's not labeled as being part of biochemistry, but I believe it is part of biochemistry. So I put in topic B6 there. It's basically a way of measuring how efficient a process is by looking at how much waste is produced, or actually looking at what percentage of the products are not wasted. And so to skip ahead, this is the equation. You pretty much just add up the molar mass of the products, including the coefficients from the equation. So it does depend on the mole ratios. And you divide it by the molar mass of all the reactants. So what you're looking for ideally is that you turn all of your reactants into desirable products. And so an example like this, in the Haber process, it has 100% atom economy because all the reactants turn into products. In this example, however, if this is your desired product here, then the HCl would be considered a waste product, and so the percentage economy would be below 100%. However, if you can use the waste products in some other process later, then it's not really a waste product, and that improves the atom economy. So like when we talked about the hydrodesulfurization of coal, hydrogen sulfide is sort of the waste product. It's not the coal that you're looking for, and so the percentage atom economy is quite low. However, if you use the hydrogen sulfide to make sulfuric acid, that improves the atom economy to 100%. And on this page are a couple of other examples of ways in which the world has gotten more green, using liquid carbon dioxide to help dissolve things out of foods is a lot safer for the food, and if it doesn't use harmful chemicals to do that, you don't have to use really harsh nonpolar solvents. The clothing industry And the cosmetics industry can use enzymes to allow them to do cleaning or production at lower temperatures, so they're more energy efficient. And bioplastics, as we talked about, are more biodegradable, and so they lead to less waste. So you can read through these lists, the list of green chemistry examples, and that is the close of the option and of the syllabus.